Welcome, everyone. I'm Karynne Patterson, an analyst from the University of Washington Center for Rare Disease Research. I'm going to talk today about how we get the data from the sequencing machines into formats that we can analyze and hopefully use to solve some rare diseases. Next slide. First, we're going to cover some background on sequencing methods. I'm going to focus heavily on DNA next-generation sequencing with Illumina short reads and calling single nucleotide variants, or SNVs, and short insertions and deletions called indels. Then I'll briefly touch on calling larger events, or structural variants. We'll go over what RNA sequencing provides. And lastly, I'll touch on the newest technology, sometimes referred to as third generation, that creates 1,000 base pair long reads. Next slide. The general workflow in next generation sequencing is to shear whole DNA into smaller fragments. Sometimes these are amplified, sometimes not. Adapters are added, and then base by base, the machine captures the AT, Cs, and Gs sequence from each fragment into what's called a read. Reads are aligned to a reference genome, and any mismatches with that reference are called as variants. Next slide. The reference sequence represents generally what's most common in humans, but it doesn't capture all variation. And so sometimes it can bias a source, as reads are penalized for not matching a reference properly. Some more computationally expensive methods don't use a reference at all and align reads naively based on overlaps, but the traditional methods rely on a reference file. As we get more data globally, references evolve. For example, the human reference GRC38 is most commonly used today, but GRC37, the previous version, is still used by many, and a newer, more complete reference with very few remaining gaps is beginning to be adopted, called T to T, telomere to telomere. Next slide. NGS workflows have three main types of the genome they capture. Whole genome sequencing, or WGS, includes the entire genome, the whole apple, if you will, coding, non-coding, and generally has equal coverage across it all. Whole exome sequencing, or WES, targets just the space that codes for proteins with specific adapters, only about 1% of a genome, the slice of the apple here, and is sparsely covered. There are high coverage peaks over exons and very little or no reads mapping to intronic or intergenic regions. The third type is specifically targeted regions or panels. Maybe you have a gene panel for the disease you're studying. Smaller regions generally get higher coverage in NGS and are cheaper, but the other side of the coin is that your variant of interest may not be in your panel or even in coding regions, or it's a more complex rearrangement that has breakpoints that need to be covered to be detected. Next slide. Raw data direct from the sequencer contains the sequences of individual reads and base qualities for every position in the read. FASTQs are text files with 4 lines per read and contain these sequence and base qualities. For millions of reads, you can imagine that these files get extremely large. Binary formats exist in BCL files that are specific to Illumina or unaligned BAM files. We then use bioinformatics software like BWA, the Burroughs-Wheeler alignment tool, to align or position these reads to our reference sequence. Next slide. Now, as these new files contain everything that was in a FASTQ, plus alignment info and scores, we need more efficient ways to store this information. Compressed binary files called BAMs are able to hold all of this and are indexed for faster information retrieval. Note, these are not text files and are most easily viewed or manipulated with a tool called SAM tools. SAM referencing the original format of a tab delimited sequence alignment file. More recently, these files have been able to be compressed even further into CRAM files. By choosing scores to drop and making reference-based assumptions about alignments, CRAMs are able to be 30 to 60% smaller than BAM files. It's called a lossy compression, and it's extremely helpful as sequencing data stores just keep increasing and increasing. Next slide. BAM format contains a ton of information, but I'm only going to highlight a few. Columns 10 and 11 contain the read sequence and base qualities, while column 3 and 4 label the genomic location that this read was mapped to. The aligner will also give the read a mapping quality and define any mismatches with the reference with something called a cigar string. Next slide. Mapping quality, or MapQ, is a log-based score of probability a read was misplaced or wrong. For example, a common threshold is a score of 20, which means the probability it was mismapped is 1 in 100. We generally think that's a pretty good probability. CIGAR stands for Compact Idiosyncratic Gapped Alignment Report. It's a shorter, more compact way to note differences from the reference than just repeating the reference itself. An 18 base pair read that maps perfectly would be noted as 18M, M for match. This read, shown here, has a 2 base pair deletion in the 3rd and 4th positions. Its cigar string is represented as 2M for the first 2 matches, 2D for the 2 deleted bases, and then 14M for the rest of the read, matching the reference. You can look up more complicated strings in the BAM specification below, but you should have a basic idea of how to read a BAM file map. Next slide. Another great tool to work with BAM files is the Integrated Genomics Viewer, or IGV, from the Broad. The tool allows a user to specify the reference and an aligned BAM and will display read-by-read coverage across the genome. In the first picture, here's displaying a heterozygous single nucleotide variant showing, and it shows which reads have the variant and which reads don't. The direction of the reads forward and reverse is denoted by blue and red colorings here. The sequence doesn't look messy. There's not a ton of mismatched calls, and the coverage is deep. I think we can be confident visually that this is a real variant. The second picture below shows the sparsity of exome sequencing compared to whole genome sequencing. Well, it's too small to see the detail of the coverage bars above the reads. The whole exome sequencing average is about 250x, while the whole genome sequencing average is about 50x. Less reads per base in whole genome sequencing, but more uniform coverage. Let's say you see a variant in IGV that isn't as clear as the one I showed you at the top. We need a variant caller to make these decisions for us bioinformatically. Next slide. We can use SNV and indel callers like GATK haplotype caller to decide when a variant should be called and when not. The programs look at how well the reads are mapped, how many reads are mapping to that location, and if heterozygous calls are balanced by equal read support, called allele balance. For indels, callers also look if an indel alignment has the same size and the same start and stop points. Next slide. Variant callers report their info out in a standard text file called a VCF file. Like BAMs, they can be compressed and indexed for easier storage and manipulation, but they're a little easier to read as a simple zcat in the command line will show you compressed VCF's output. VCF tools and TabX can also be useful tools for working with VCFs. VCF's format starts with a header of metadata denoted with 2 pound signs or hashtags starting each line. The last line of the header has 1 hashtag and is the column headers for the variant lines below that. It also includes the IDs of any samples in the VCF. As you can see, the blue variant box at the bottom contains quite a bit of information. What is contained in the info and format sections of a VCF can change, but each field should be described in the metadata header if you need help for referencing. This line in the blue box showing a SNV change from a base T from a C in the reference. The genotype call is heterozygous with 0 representing the reference call and 1 representing the alternate call of a T. AD in the second field stands for allele depth. So 21 and 20 are representing the 21 reads with the first, in this case, reference allele, and 20 reads representing the second, the alternate allele. Genotype GQ stands for genotype quality, which is the difference between the most likely genotype and the next most likely genotype. A score of 99 means the caller is very confident in this call. Next slide. Here's another diagram of an entire VCF, complete with a header and VCF body sections. We see the red mandatory header lines and some green optional descriptions. Next slide. In this section in the header, highlighted here, matches the fields in the info and format fields below. So we've got a fully documented file. It's also considered a multi-sample because there's more than one sample column noted here. Next slide. Zooming in on the variant lines, the first variant is a single nucleotide variant change of a G from an A. Looking to the right at the genotype, the first sample, NA12878, has a heterozygous variant represented by the genotype 0 and 1. The second sample, NA12879, is a homozygous reference at the site denoted by 00. Next slide. The last tidbit I wanted to touch on in this example is the third variant here at the bottom right over here, a 1 base pair deletion of a G. The reference base before it is included to help denote the location. Insertions are often documented in a similar form. Next slide. Rather than write every base of the genome out, which would make enormous text files, but also wouldn't make sense for a sparse exome sample, only covering 1% of it, VCFs often report only variant sites. This gets complicated when you merge samples and don't have information on sites where some samples are referenced. Enter the genomic VCF. Contiguous regions of all reference non-variant sites can be condensed into one line in this file. Callers will include an average depth and scores over this region, so we have more confidence that this region is non-variant than assuming with only missing data. The first line in blue is a reference block from 10,204 to 10,210 with over 30x in coverage. Then there's a variant at 10,211, and the line after that picks up at the next base, 10,212 to 216 as another reference block. We're able to collapse 13 bases into 3 lines here, and GVCFs are even able to do this on much bigger regions of references. Next slide. As I mentioned, we need full genotypes when working with multiple samples, especially in family studies. A de novo invariant is a variant present in a proband or child that is not seen in either parent. If we included a variant-only VCFs in our combined step, we'd have missing data for the parents at this site, represented by dot/dot. That's how you denote missing data in a VCF. And we'd only be left with a single SNP in the proband with no more information. This doesn't work. By leveraging GVCFs in the info in their reference blocks, we would be able to accurately genotype the parents as homozygous reference, and this child is having a spurious de novo SNP. Next slide. GATK's genotype GVCFs does this joint calling process from GVCFs, so we can have a squared off multi-sample with no or very little missing data. In addition, joint calling helps booster low frequency variant calls and has a greater power to filter out false positives. Utilizing pre-computed GVCFs is also much less computationally expensive than joint calling straight from BAM files. Joint calling can also benefit from filtering, especially with GATK's VQSR pipeline called Variant Quality Score Recalibration. It has a machine learning algorithm that includes known variation with your sample set and provides a confidence level of your choosing. It's definitely a helpful place to start with passing these filters when moving along to variant analysis. Note, smaller cohorts should stick with the standard threshold cutoffs rather than using this machine learning method. Next slide. So what I've talked about so far mostly relates to small variants under 50 base pairs. Structural variants generally are a term that covers any genetic rearrangements over 50 base pairs. You often can see these large events in IGV, like the picture at the bottom on the right, as there's literally no reads spanning this region in the middle. It's likely a homozygous deletion. Not all structural variants can be seen so clearly in IGV, but it's a great place to evaluate any structural variant calls you have. And structural variant callers, there are a lot of programs. Most utilize BAM files and rely on read depth, split read, and read pair methods to detect and genotype these events. Because of the different types of evidence and the biases inherent to each caller, it's often recommended to use more than one caller in your analysis, as no one program is especially great at calling structural variants. Next slide. NGS sequencing is also really great for working with RNA. RNA-seq allows you to identify expression outliers or aberrant splicing, whether you start with a DNA variant and want to see its specific effects, or you start with RNA and need a place to look for any changes in the genome. The workflows in RNA are similar to DNA, starting with reads and ending with variant calls, but software and metrics for RNA are different. Next slide. And the last thing I wanted to go over was the latest technology, often labeled as third generation sequencing or just long read sequencing. Whereas NGS is limited to read lengths of a couple hundreds, long reads give read lengths of thousands. The specificity allows mapping of complex or repetitive regions that short reads just don't have the uniqueness or span to cover. The drawbacks are that the error rate of these bases in long reads are much higher, and it costs much more to get the coverage needed to balance the error rate. Technological improvements are making base calling better, and the bioinformatics software that uses these technologies is in active development. We'll see a lot more with long reads in the future. Next slide. In summary, today we mostly focused on calling SNVs and short indels with Illumina short read NGS technology. We walked from raw data from the sequencers into binary alignment files called BAMs, and we saw how they can be visualized in a tool called IGV. And then we went over VCFs, GVCFs, and our final variant called product, a joint called VCF file. We then touched on calling larger structural variant events in NGS, and we added some info on RNA-seq. Then we looked to the future for long read sequencing that's rapidly evolving and bringing so much new data to our field. Next slide. Thanks so much for the opportunity to speak to you all today. And I wanted to thank my colleagues at the UW-CRDR and in the Nickerson lab for their help in preparing this talk.